Welcome back to the deep dive. Today, we're doing something kind of different. We're diving into the world of theoretical neuroscience.
Oh, cool.
Yeah, so we're cracking open Theoretical Neuroscience by Peter Dayan and L.F. Abbott to try to understand how our brains make sense of, well, everything.
Ambitious.
Very. We're basically talking neural code breaking. You know how those tiny neurons in your head take like sights, sounds, and turn them into thoughts and actions?
Like deciphering an alien language.
Yeah.
But the aliens live inside our own skulls.
Right. I love that. So the book jumps right into this idea of neurons using spikes to communicate. Not actual spikes, of course.
No, we're talking electrical signals. Action potentials, they're called. Each spike is like a blip, a signal on the neuron's radar. And these blips form patterns that represent information.
So it's not just a neuron being on or off, but like the timing of these spikes. That matters too.
Exactly. Timing is key. Think of Morse code. The information is encoded in the sequence and timing of those dots and dashes. The spike trains work similarly. The frequency, the pattern, all of it conveys different messages.
Okay, so we've got these spike trains and the overall firing rate of a neuron. But then the book mentions this inhomogeneous Poisson process, and honestly, it sounds like a drink order at a fancy coffee shop.
It does, doesn't it?
Yeah.
But it's a little less glamorous than it sounds. A mathematical model basically trying to explain how random these spike trains are.
Random, really?
To a degree, imagine you're watching shooting stars. The timing of each one seems random, but there might be bursts of activity, like, during a meteor shower.
Oh, okay, I get it. So
Yeah.
a neuron might be more likely to fire during certain events, but the exact timing of each spike could still be sort of random.
Yeah, you got it. This model has been pretty helpful, especially for understanding some basic stuff about how neurons behave. But it's not a perfect picture. Real neurons are way more complex than just random spike generators.
Right, of course. So then how do scientists even begin to decode these signals? How do they figure out what makes a neuron fire?
It's a tough job, but they use a technique called reverse correlation. Kind of like figuring out a lock by trying different keys until one fits.
Yeah.
Researchers give the neuron random stimuli, like white noise, if we're talking about auditory neurons, and then they see what triggers a spike.
So they're basically like throwing information at the neuron and seeing what makes it tick.
Yeah, pretty much. And by looking at that relationship between the random stimulus and the spike times, they can map out the neuron's receptive field.
Receptive field.
It's like the neuron's area of expertise,
Yeah.
the specific type of information it's most sensitive to.
So a neuron's receptive field could be anything, like sounds, visual patterns, even, I don't know, abstract concepts.
Absolutely. And it's not just about figuring out what it responds to, but understanding how these receptive fields are organized in the brain. That's the really interesting part.
So it's like piecing together a puzzle, seeing how each individual neuron with its own receptive field fits into this big picture of how we perceive the world.
Exactly. And it's that puzzle, that intricate web of connections that theoretical neuroscience tries to unravel.
Fascinating stuff. We've covered a lot of ground already, but we're just getting started. Up next, we'll be taking a closer look at how these receptive fields actually work, especially in our visual system. Stay tuned. Okay, so now we're going to journey into the visual system. This is like one of my favorite parts of the brain. It's just amazing how it all works.
It really is quite something. Taking something as fundamental as light and transforming it into, well, our entire visual world.
It's mind-blowing. Okay, so let's start at the beginning.
Yeah.
With the retina, the very back of the eye.
Right. That's where light first gets converted into those neural signals we were talking about. And it's where we find these special neurons called retinal ganglion cells.
Retinal ganglion cells.
Yeah, they're like the gatekeepers of visual information. They send the signals from the eye up to the brain.
And they have those center surround receptive fields we talked about before. Right?
Exactly. And those receptive fields, they're not just passively relaying information. They're already doing some pretty heavy lifting when it comes to processing visual information.
Can you remind me how those receptive fields work again?
Sure. Think of it like a donut.
A donut, okay.
Yeah, so the center of the donut hole, that's the center of the receptive field. Now, when light hits that center, it can either excite the neuron, making it fire more spikes, or it can inhibit it, making it fire less.
And the surrounding ring, the actual donut part, does the opposite.
Exactly. So you've got this push-pull relationship going on, and it's super sensitive to differences in light intensity.
So it's basically like a built-in edge detector.
Yeah, you could say that.
Huh.
It helps us see where one object ends and another begins.
So even at this early stage, in the eye itself, the brain is already starting to analyze the visual world.
Oh, absolutely. But it gets even more complex as that information travels from the retina up to the visual cortex. That's where we find these neurons called simple cells.
Simple cells.
Simple, in a manner of speaking. They have receptive fields that are tuned to very specific orientations of light.
So while those retinal ganglion cells are looking for edges and contrast, these simple cells are looking for, like, lines and angles.
Precisely, like little tiny edge detectors, each one specialized to respond to lines of a particular orientation, like vertical, horizontal, diagonal, and so on.
Wow. It's like our brains are deconstructing the visual world into lines and edges, like an artist sketching out a scene.
It's a great analogy.
Yeah.
But it doesn't stop there. Then we've got complex cells, which add another layer of complexity.
Complex cells, how are they different?
Well, they're kind of like simple cells in that they respond to those bars of light at specific orientations, but they also care about the direction of movement.
So a complex cell might be tuned to, say, a diagonal line moving upwards, but it wouldn't fire as much if the line was stationary or moving downwards.
That's exactly right. And this allows us to perceive motion and form with incredible precision. It's really quite remarkable.
So we've got this hierarchy of visual processing with each type of cell building on the one before it.
And all of this is happening in parallel. You've got different populations of neurons all analyzing different aspects of the scene at the same time. Color, motion, depth, it's all being processed simultaneously.
It's like an orchestra with all these different instruments playing together to create this unified visual experience.
I like that. A symphony of neural activity. But here's the thing. Up until now, we've been talking about individual neurons. But to really understand vision, we need to zoom out and look at how populations of neurons work together.
This is where that population decoding comes in, right?
Exactly. It's about understanding how the activity of many neurons combines to represent information. You see, each neuron might be tuned to a slightly different feature, but by looking at the collective activity, we can get a much richer picture of what's going on.
I get the idea, but can you give me an example? It's kind of hard to imagine how something as complex as, well, seeing could emerge from the activity of so many individual neurons.
Sure. Let's take a look at cricket circle interneurons.
Cricket circle interneurons.
They're these neurons that are responsible for detecting wind direction. Each neuron is like a tiny little wind sensor tuned to a specific direction.
So each neuron has its own preferred wind direction.
Right. And when the wind blows from that direction, that neuron fires a bunch of spikes. It's basically shouting, hey, wind's coming from over here.
I love that analogy. And then the brain, it listens to all these neurons shouting and it figures out which direction the wind is coming from.
Exactly. It analyzes the activity of the whole population, like taking a vote. The neurons that are firing the most, they win, and that determines the perceived wind direction.
So even though each neuron only responds to a narrow range of wind directions, the population as a whole can represent a much broader spectrum.
And it's super efficient, which is important because the brain needs to conserve energy. It can't afford to have neurons firing all the time for no reason.
Right, of course. So this population coding, it's not just for crickets, right?
Oh, no. It's thought to be fundamental to how our brains work, too. Everything from recognizing faces to understanding language probably relies on this principle.
This is all so cool. We've gone from spikes to receptive fields to populations of neurons. Where do we go next?
Well, now that we have a sense of how neurons represent information, I think it's time we talk about information theory and how it can be applied to the brain.
Information theory. That sounds kind of intimidating.
It might sound complicated, but it's basically a way to quantify how much information is transmitted by a signal.
So in this case, we're talking about how much information is carried by those spike trains.
Exactly. And one of the key things we've learned from information theory is that a good, efficient neural code should maximize the amount of information transmitted while minimizing the metabolic cost.
That makes sense. The brain is a very energy-hungry organ, so it wouldn't make sense to waste energy on an inefficient code.
Right. And this is where things get really interesting, because it turns out that the structure of receptive fields, like those center surround receptive fields we talked about in the retina, they're actually optimized for extracting information from the kinds of images we typically encounter in the real world.
Oh, wow. So the statistics of the visual world, they've actually shaped the wiring of our visual system.
Exactly. Evolution and experience have shaped receptive fields to maximize the amount of information they can extract from the environment.
So our brains are basically adapted to the specific structure of the world we live in. That's pretty amazing.
It is, isn't it? And it's just one example of how information theory can give us insights into how the brain works. It's a powerful tool for understanding the principles of neural coding.
This is really blowing my mind. I had no idea how much theoretical work goes into understanding something as, well, seemingly straightforward as vision.
And we've only just scratched the surface. There's still so much more to explore. But now that we have this foundation in neural coding and in information theory, I think it's time we move on to computational neuroscience.
Computational neuroscience, what's that all about?
It's about using computers to model and simulate the activity of real neurons.
So we're going from observing neurons to actually building virtual versions of them.
Exactly. And these models are like building blocks. We can use them to construct virtual circuits and test different hypotheses about how the brain works.
It's like having a virtual lab for experimenting with the brain.
That's a good way to put it. And there are all sorts of different models, ranging from very simple to incredibly complex.
Like those integrate and fire models we talked about earlier.
Exactly. Those are a good example of a simple but powerful model that can capture a lot of the basic behavior of neurons. But there are also more complex models, like the Hodgkin-Huxley model.
And that one goes into more detail about how the spikes are actually generated.
Yeah, it delves into the biophysics, the flow of ions across the cell membrane.
It's amazing how these models can bridge the gap between, the biology of neurons and the computational processes of the brain.
It really is. And by using these models, we can not only understand how the brain works, but we can also simulate what might happen if we change things.
Like test out different hypotheses about how different brain regions interact.
Exactly. Or how learning and memory might work at the neural level. We can even explore potential treatments for neurological disorders. The possibilities are really quite vast.
This is all so fascinating. It really feels like we're on the cusp of some huge breakthroughs in our understanding of the brain.
I agree. And it's all thanks to this amazing interplay between theory and experimentation. That's what drives progress in neuroscience.
Okay, well, I think we've covered a lot of ground for today. From the basic language of neurons to these amazing computational models that are helping us unlock the brain's secrets. It's been an incredible journey.
It has indeed. And I want to thank everyone for joining us on this deep dive. I hope you'll continue to explore the fascinating world of theoretical neuroscience.
Absolutely. There's still so much more to discover. So to all our listeners out there, keep those neurons firing, and we'll catch you on the next deep dive. All right, so last time we were talking about model neurons. You call them Legos for the mind, which I gotta say is a great way to think about it. We were talking about those integrate and fire models and, the neuron as a leaky bucket.
It is a helpful analogy, isn't it? It gets the core concept across. The neuron accumulates all this electrical input, and then when it hits a certain threshold, bam, it fires a spike.
Yeah.
And then the process starts all over again.
And you were saying that even though it's a simplification, it can actually explain a lot about how neurons behave.
Yeah, it's been surprisingly effective. But then, of course, there are those more complex models, like the Hodgkin-Huxley model we touched on.
Right, right. So if the simple models work so well, why even bother with the more complex ones?
Well, think of it like building a model car. You can start with a simple one made of blocks, gives you the general shape, the basic idea. But to really understand how the engine works, you need a more detailed model.
Right. So the Hodgkin-Huxley model is like looking under the hood of the neuron.
Exactly. It gets into the nitty-gritty details, the biophysics, like the actual flow of ions across the membrane, the stuff that actually creates the electrical signals.
Wow. It's amazing how these models connect the biology of the neurons to those computational processes we've been talking about.
That's really the heart of computational neuroscience.
Hmm.
It lets us study the brain at so many different levels, from those tiny ion channels to huge networks of neurons.
And this means we can use these models not just to understand how the brain works, but also to try and predict what will happen if we, mess with things a little.
Yeah, we can test hypotheses, see how different parts of the brain might interact, or how learning and memory work at the level of neurons. Even explore ways to treat neurological diseases. It's a powerful approach.
So it's like having a virtual brain to play with.
In a way, yeah. But we have to remember that all models are simplifications. They're not perfect representations of reality.
Right, of course. It's like using a map to navigate a city. The map is useful, it helps you get around, but it doesn't show you every single detail.
Precisely. We have to keep the limitations in mind and not read too much into the results. But that said, computational neuroscience has already helped us learn so much about the brain.
For sure. We've learned so much in this deep dive alone. We started with the basic language of neurons, then explored these sophisticated computational models that are helping us make sense of it all. It's been a wild ride.
I agree. And it's just the tip of the iceberg. There's still so much we don't know about the brain.
Absolutely. But the future of brain research is looking bright. With all the new tools and techniques we have, who knows what we'll discover next?
I can't wait to find out.
Well, on that note, I want to thank everyone for joining us on this deep dive into the world of theoretical neuroscience. I hope you've enjoyed it as much as we have.
It's been a pleasure.
And until next time, keep those neurons firing.